I am Pascal Cossard from the Pasteur Institute in Paris, and I am also a senior international research scholar from the Howard Hughes Medical Institute. What I would like to do is to share with you how we study the bacterial pathogen Listeria monocytogenes to address key issues in infection biology, but also in fundamental microbiology and in cell biology. So my talk will be divided into 3 parts. In the first part, I will be rather general and focus on bacterial pathogenesis, giving you an overview of the Listeria paradigm. In the second part, I will show you how the study of Listeria monocytogenes has really provided key new concepts in fundamental microbiology and really participated to the renaissance of this discipline. In the third part, I will focus on the cell biology of the infectious process and show you how the study of this infectious process has unveiled new components, new mechanisms, which are taking place in the mammalian cells. So what is microbiology? Microbiology is these disciplines which addresses organisms which are not visible with the eye. So these organisms are about 50-fold smaller than mammalian cells. They are the size of organelles which are in mammalian cells, like mitochondria. So bacteria are very diverse. Bacteria are very, very rarely pathogenic. There are many bacteria in the environment, like Streptomyces, which is producing the antibiotic streptomycin. There are bacteria which are in the environment but live in symbiosis with, for example, the roots of the vegetables, like Cynorhizobium meliloti, which helps the bacteria to capture the nitrogen. There are bacteria which are commensals, and you all know that we have bacteria in our intestine. And finally, there are those which are the pathogenic bacteria, like Shigella flexneri, or the bacterium that I am going to talk about today, which is Listeria monocytogenes. So pathogenic bacteria can be classified in several categories. I think there are 2 main categories of pathogenic bacteria. Those which are acting by producing a single potent toxin, like Corynebacterium, which produces diphtheria toxin, or Clostridium botulinum, which produces botulinum toxin. But very often, pathogenic bacteria are using multi-factors, like adhesion, colonization, tissue destruction. So all the bacteria that I'm showing you here, Pseudomonas, enteropathogenic E. coli, Staphylococci, are mostly extracellular bacteria. But there are intracellular bacteria. And among the intracellular bacteria, I see 2 categories. Those which are intracellular because they resist killing by professional phagocytes, and those which are inside mammalian cells because they are active, they can enter into cells which are normally non-phagocytic. For example, Listeria monocytogenes, my favorite organism, is able to enter into cells which are normally non-phagocytic. So Listeria was not discovered by Pasteur. It was not discovered by his competitor Robert Koch. It was discovered by Murray in England in 1926. I consider that there are several important dates in the Listeriology. So in the 60s, Macanese discovered that recovery from infection by Listeria or protection against a secondary infection is not due to antibodies, but is due to a T-cell response. And this was the beginning of the use of Listeria as a tool to study the induction of a T-cell response. Then, in the late 80s, several groups in the world started to combine molecular biology, bacterial genetics, and cell biology to address the virulence of Listeria. So this was the beginning of a new discipline that we call cellular microbiology. And this is still going on. Then, in the beginning of 2000 of this century, the genome of Listeria monocytogenes was sequenced, and this was the beginning of a large number of studies, which we can call postgenomics. And very recently, we have shown that Listeria is inducing epigenetic changes, and this is really contributing to what we call epigenetics and infection. So the genus Listeria is growing. It has grown recently. It contains several species, including 2 which are pathogenic. Monocytogenes is pathogenic for humans and animals, and Listeria ivanovi is pathogenic only for animals. So Listeria monocytogenes is a gram-positive bacterium which is ubiquitous in the environment. It's a saprophyte. It can live on decaying vegetation, and it's a pathogen, again, for humans and animals. It's a bacterium which is motile at low temperature, and more importantly, it can replicate in the cold at high salt concentration, at low pH, those conditions which are used generally to conserve food in the refrigerator or in factories. So this is the bacterium which can contaminate the food product, and it is an economical problem when food is contaminated. And really, in that case, food is recalled from the market, and it has led to dramatic economical situation. So what are the successive steps of human listeriosis? Well, very simple. You ingest contaminated food product, the bacteria reach the intestine, and then the bacteria can cross the intestinal barrier, reach the liver and the spleen, and then via the blood, they can disseminate to the brain or, in pregnant women, to the fetus. So it's a really dangerous bacterium, but the disease only occurs in certain cases, which are immunocompromised individuals, old people, very young babies, or pregnant women. At the cellular level, the bacteria enter into cells, as I said previously. Then they are trapped in some kind of a vacuole, which is very rapidly lysed. The bacteria escape from the vacuole, and they start moving. Not only they are moving inside one cell, but they can go from one cell to the other. When they arrive to the other cell, they escape from the two-membrane vacuole, and they start to multiply again in the second infected cell. So I would like to briefly discuss the entry process. You see that when a bacterium is entering into a cell, the membrane has to rearrange, and the cytoskeleton, which is under the membrane, has also to rearrange. We have studied that into details. So first, we were interested to know what are the molecules of the bacterium which are important for this entry. So we isolated a mutant, a transposon mutant, which was inserted in the chromosome upstream of the 2 genes, ENLA and ENLB. So this mutant was not invasive. Then we deleted each of the 2 genes, and we could show that, in fact, the first gene, ENLA, which encodes internalin, is used for the entry into epithelial cells. The second gene, when deleted, the second gene is used for entry in all human cell types. So let's focus on the first one. The first one is encoding a protein which is called internalin. Internalin was shown to interact with a protein which is called E-cadherin. E-cadherin is this protein which is used for the interaction between epithelial cells together. So E-cadherin binds to E-cadherin and mediates the binding of epithelial cells to another epithelial cells. Well, Listeria is using the property of E-cadherin to enter into cells. What is interesting is that internalin interacts with human E-cadherin, but cannot interact with a mouse E-cadherin. And this species specificity is due to a single amino acid at position 16, as we have been able to show. So internalin cannot interact with the mouse Ikaderin. And this was really a problem for us because we wanted to test what is the relevance of the interaction between internalin and Ikaderin, and we wanted to test the relevance of this interaction in a mouse model. So we decided to create a transgenic mouse which would express the human Ikaderin at the level of the intestine. And I'm going to show you the results of the analysis of the infection in normal mice and in transgenic mice, and show you that, in fact, the interaction between Ikaderin and human, of internalin and human Ikaderin, is absolutely critical for listeriosis. So on the left, you see the results of the infection of wild-type mice. On the right, you see the infection with the human Ikaderin transgenic mice. So if you infect wild-type mice with 5 to the 10 bacteria, 10 to the 10 bacteria, there is 0% mortality. If you infect with the same amount of bacteria with the mutant, there is 0% mortality. But if you infect orally human Ikaderin transgenic mice with 5 10 to the 10 bacteria, you see that there are 100% mortality. If you infect with the internalin mutant, the delta INLA mutant, there is 0% mortality. This key experiment is really demonstrating that the interaction between Ikaderin and internalin is critical for a productive Listeria infection, which leads to lethality. So this was a big step, but of course, now the work is to really understand how Listeria is interacting with Ikaderin at the level of the intestine. I'm going just to show you how my colleague Marc Lecuy is now pursuing this work and show you that, in fact, it's clear that Listeria is associated with human Ikaderin, which is present on the intestinal villi, and about 20% is associated at the tip of the villi, and 90% is associated at the border of the villi, on the side of the villi. Okay, I told you about the internalin interacting with Ikaderin. I told you that this is an interaction which is species-specific. What about the second gene which is involved in entry? Well, the second gene is encoding a protein which is called ENLB, and ENLB interacts with a receptor which is ubiquitous, which is the hepatocyte growth factor receptor, which is called MET. So ENLB interacts with MET in all cells, including those cells where there is or there is not Ikaderin. An interesting result is that there is also a species specificity for ENLB. And in fact, in contrast to ENLA, which interacts with human Ikaderin but does not interact with the mouse Ikaderin, ENLB interacts with the human MET and also interacts with the mouse MET. And we have been able to show, together with Marc Lecuy, that in fact, ENLA and ENLB act in concert to cross the placental barrier. So I will not talk more about the entry. The entry is thus mediated by ENLA and ENLB. We have been able to decipher other genes which are involved in the process of infection. So I already mentioned listeriolysine O as a protein which is a key protein for the escape from the vacuole. I would like to focus on a protein which is able to propel the bacterium inside cells. So the phenomenon of actin-based motility was discovered by Tilney and Portnoy. And as shown on this movie from the Julie Theriot lab, you see that the bacteria are really moving inside cells. We have been able to show that this movement is mediated by a protein which is called ACT-A. And you see on the left part of this slide that in contrast to the wild-type bacteria, this ACT-A mutant is not able to produce these little comet tails that you see on the movie. So this ACT-A mutant does not produce a protein which is on the surface of the bacterium and which propels the bacteria inside cells. So the bacteria are still alive, they multiply inside cells, but are not able to move, they are not able to spread from cell to cell, and the mutant is highly attenuated. What is interesting concerning all these virulence factors, I mentioned the Listeria O, which is encoded by HLY, I mentioned ACT, I mentioned ENLA and ENLB, is that all these virulence factors are co-regulated by a single protein, PRFA. And in my second talk, I will show you that this PRFA protein is really, really tightly regulated. So one big step, as I said in my introduction, one big step in the listeriology was the elucidation of the genome of Listeria monocytogenes. So the sequence was performed in the same time that the sequence of Listeria innocua was performed, and Listeria innocua is non-pathogenic. So the goal of this project was to get two genomes and to be able to compare the genome of the pathogenic and the genome of the non-pathogenic species in order to find new virulence factors. And in fact, we found some of them. I'm going just to give you two examples. For example, we found that there were many genes, and by other techniques, we were able to show that they were regulated also by PRFA. And I would like to focus on the last gene of this slide, which is BSH. This was a surprise. What is BSH? BSH is a bile salt hydrolase. We were not expecting to find such a gene in Listeria monocytogenes, and this gene is absent from Listeria innocua, as you see by the yellow color, which is not present in Listeria innocua. So what is the bile? The bile is a complex liquid, which is made of cholesterol-derived compounds. And the role of the bile is to emulsify the lipid from the diet, from the food, and it really also acting as an antimicrobial agent. So the bile is made in the liver, is stocked in the gallbladder, and it goes in the intestine after the meals. So the commensals, the intestinal commensals, have evolved several tricks to resist the bacterial action of the bile salt, including the production of a bile salt hydrolase. So we were wondering whether this bile salt hydrolase was playing a role in the virulence of Listeria, or at least in the persistence of Listeria in the intestine. So we created a mutant, and we analyzed the behavior of this mutant. As you can see here, compared to Listeria monocytogenes wild type, which is black, the mutant, which is in blue, does not persist in this intestine several hours after the infection. So clearly, the BSH is critical for Listeria persistence in the intestine. So similar to commensals, pathogenic Listeria counteracts the antibacterial activity of bile salts by the BSH. And this allows the persistence in the intestine and is critical for virulence. Another result that we got from the genome was also a surprise. At the time, we and others were studying the role of the peptidoglycan in the innate immune response. And it was a surprise that the Listeria peptidoglycan was stimulating poorly the innate immune receptor node. And we wonder whether this peptidoglycan was maybe modified. And to make a long story short, the answer is yes. And we found that, in fact, there is a deacetylase in the genome of Listeria, which is acting on the peptidoglycan. So we created a mutant, and we could show that the deacetylase mutant is much more sensitive to lysozyme, which is an enzyme which is cleaving the peptidoglycan. More than that, we could show that the growth of the deacetylase mutant is attenuated in mice. So compared to the wild type, the PGDA mutant is really destroyed very rapidly. So in conclusion, Listeria is able to deacetylate the peptidoglycan through a peptidoglycan deacetylase to evade innate immunity. And the mutant, the PGDA mutant, is really strongly attenuated in virulence. And we could really show that the peptidoglycan is deacetylated in the wild type and is not deacetylated in the mutant. So the conclusion of all that is really that Listeria is using a variety of strategies. I've already mentioned ENLA interacting with Ikaterin. I've already mentioned ENLB interacting with the growth factor receptor, which is called MET. I have mentioned ACT, which is used for actin polymerization. I just mentioned the PGDA, which is a peptidoglycan, which is a gene involved in peptidoglycan modification to evade the host immune response. There are a plethora of strategies used by Listeria, which are described in the two other talks. For example, we have shown that, or it has been shown that ACT not only is used for actin polymerization, but also for escape from autophagy. We have shown that there is the PGDA, which is modifying the peptidoglycan, but we found that there is also an acetyltransferase, which is modifying the peptidoglycan, again, to evade from the innate immune response. This listeriolysine O toxin, which I briefly mentioned, which is used for the escape from the vacuole, seems to have many roles during Listeria infection. It makes pores in the membrane, and this induces a series of events like mitochondrial fragmentation, desumylation of proteins in the mammalian cells, also histone modification. But all these will be described in my third talk. So I think at this stage, I would like to say that Listeria monocytogenes has really emerged as a multifaceted model in different areas and as a reference. For example, in microbiology, I will talk about gene regulation by non-coding RNAs. I've just mentioned cellular microbiology. In cell biology, Listeria has been used to really study actin-based motility, endocytosis, cell adhesion and signaling, mitochondrial dynamics, post-translational modification. Listeria has been one of the first organisms used as intensively for comparative genomics, for whole genome, transcriptomic, and now for proteogenomics. I mentioned briefly that Listeria is able to also target the chromatin, and really, I think the field of epigenetic and infection will benefit from studies which have been carried in Listeria. I also talked about the immunology. I told you that Listeria was shown that recovery from a Listeria infection is due to a T cell response, and really the immunologists are really using Listeria as a tool for the induction of for the study of the induction of the T cell and the innate immunity. And also, Listeria is used as a live vaccine for cancer therapy. Finally, I told you that Listeria is able to cross the human type barrier that are the intestinal barrier, the placental barrier, and there has been very few bacteria for which it has been shown, like we have shown, how bacterial protein is critically involved in the crossing of the barrier.